Welcome. Thank you so much for tuning in. We're pleased to have you join us for the August 2019 California Current Acidification Network Roundtable discussion. The title of today's presentation is Understanding Acidification Risks Across Habitats Through a 10-Site Intertidal Network. This series is being hosted by the California Current Acidification Network. In short, the series is intended to create a dialogue among industry members, natural resource managers, and scientists within the California current ecosystem. With awareness of and access to the research and its applications and uses, the participants will be able to collaborate to better understand and adapt to ocean acidification moving forward. I'm Terry King, a member of the CCAN webinar committee, and I'll be moderating and running the logistics of today's session. During the presentation, attendees will be in listen-only mode. You're welcome to type questions related to technical issues or questions for the presenter in the questions box at the bottom of the control panel on the right of your screen. We will be monitoring incoming questions and will respond to them or pose them to our speaker after their presentation. We are also recording this session and will share the recording on the California Current Acidification website. We're very excited to have Dr. Michael Horwith with us today. Dr. Horwith leads research on climate change and ocean acidification for the Washington State Department of Natural Resources in the Aquatics Division. He oversees the Acidification Nearshore Monitoring Network. Micah collaborates with the university and tribal scientists, nonprofit organizations, government agencies, and passionate volunteers. He received his PhD in biology from the University of Washington in 2011. I'll hand the screen and the microphone over to Micah now. Welcome, Micah, and thank you for your presentation.
Hi, everybody. Thanks for joining this webinar. This is the first webinar I've ever given. And I'm going to talk to you today about a program at the Washington State Department of Natural Resources that oversees a 10-site intertidal network where we're investigating a lot of fundamental and practical questions about ocean acidification and climate change. So some of the things in this photo here are going to become clear with time. And before going into the data and the scientific findings of the network, I'm going to first give sort of an overview of its origins. So why is the Department of Natural Resources interested in acidification to begin with? Everyone in this webinar, I think, is pretty familiar with acidification as a phenomenon and its risks, the risks that it may pose to shell-forming organisms. And bivalves are really important shell-forming organisms in Washington State and the Pacific Northwest. So as an example, this is a geoduck clam for sale in a market in East Asia. And this is one destination that those geoducks have and one part of their importance. Aside from their economic value, geoducks and other shellfish are ecologically, culturally, and recreationally important. In Washington State, they're of particular importance because the industry here is so well developed and larger than in any other state, accounting for all the different species that are cultured on Washington tide flats. For DNR in particular, geoducks are very important because the wild stock fishery, the ducks that are in subtidal habitats, the harvest rights to those geoducks are auctioned off and generate revenue for the state, which goes to support natural resource management and other DNR priorities. So once acidification was articulated as a problem through all of the great leadership many of you showed on this call, DNR identified that there should be work done on this in-house, and I started work there to figure out what research gaps could be filled by work at DNR. So one of those gaps, when Washington State convened many experts to create the Blue Ribbon Panel on Acidification in 2012, was a recommendation to study the possibility of phytoremediation, or remediation by vegetation. And what does that mean as a key early action? It is pretty simple. Any underwater plant, the proposal for phytoremediation is that when that plant or any primary producer photosynthesizes, it absorbs carbon dioxide. And as a result of absorbing the carbon dioxide, changes the local carbonate chemistry. So it has effects on the local environment in ways that might matter to shell-forming organisms. When this was proposed, there was really very little work on the subject. And DNR was poised to contribute something here because there is a long-standing body of work at the agency in studying seagrass in the Salish Sea in Greater Puget Sound. So this is through the Submerged Vegetation Monitoring Program, which dates back to 2000. I think that's the right year. And ever since 2000, there's been consistent monitoring of the extent of eelgrass at many different sites. And this is the most recently generated map of seagrass extent. So DNR already had a lot of information about where aquatic vegetation is in Washington State. And the next step was figuring out whether that vegetation was doing anything to locally modify chemistry in ways that might matter for sensitive animals. And that led, after some experimental work, to the establishment of the Acidification Nearshore Monitoring Network, which now includes 10 sites all around Washington State, one on the outer coast and the rest in the Greater Puget Sound area. And there are a bunch of interlocking parts to this program. The basics of it are environmental measurements at all of these sites. So at each of the 10 sites, there are measurements of pH, temperature, dissolved oxygen, chlorophyll, and salinity taking place year-round. There is also data collected on biological aspects of the nearshore environment. So we're measuring the recruitment of shellfish, oysters, clams, and mussels. And we're also embarking on a new effort to look at the presence of geoducks across the network, especially larval geoducks through eDNA methods. And in order to understand how plants might affect carbonate chemistry, we also are measuring their characteristics. So the density of the eelgrass at all of these different sites and the morphology of the plants. And to zoom in a little bit about how this is accomplished, these are the instruments that are at all of those 10 sites that are taking measurements. The frequency of the measurements is fairly high. They're every 10 minutes. And the sensors that you see here are a mixture of commercially available instruments. I won't advertise for them right now, but we're measuring chlorophyll, salinity, pH, and dissolved oxygen across these 4 different devices. And all of those instruments are also measuring temperature. So when we put those in the water, these are intertidal deployments, which is a crucial part of the program. You can see here, we attach the sensors to these robust PVC frames and put them into intertidal environments. So this was an effort to complement the great work that's being done to monitor marine chemistry in deeper water by NOAA and others in moored stations. So we have anchors in the sediment at all of our 10 sites that are holding these instruments and taking measurements about 6 to 8 inches off of the bottom. And the depth of these deployments is between -1 and -3 feet, mean lower low water. And most crucially, at every one of the 10 sites, we have 2 stations. One of those stations is inside of eelgrass and the other is outside. And with these high frequency measurements and co-located biological observations, we're really trying to get at the mechanics of what eelgrass and potentially other aquatic vegetation does for CO2 sensitive animals. So whether it matters for an oyster, for instance, to be on the right side of this picture in a seagrass meadow or on the left side, right outside of it, we're trying to understand that. And a slightly newer aspect of the program, which I'm really been pleased about, is that we have a wonderful assortment of volunteers across the whole network who are called site guardians. And our site guardians are local partners to us who take on the responsibility of caretaking their local site. And especially in the summer, that means a lot of work, because one of the primary obstacles to collecting good environmental data in an intertidal environment is biofouling. And in the spring and summer, biofouling is happening really fast between barnacles, algae, and other fouling organisms. And our site guardians go out every low tide cycle to clean the instruments and make sure that the data they're collecting are reliable. You can also see pictures here of site guardians taking biological measurements, measuring the length of eelgrass leaves, counting eelgrass density, and taking measurements of shellfish as well. So what are the site guardians doing this summer? One ongoing effort is to understand shellfish recruitment. So this is a pretty basic hardware store method where we put out ceramic tiles at all of our 10 anemone sites inside and outside of seagrass. And we leave the tiles out there for 2-week intervals to get a snapshot of shellfish reproduction. After 2 weeks, we pull the tiles and we look at them under a microscope and count the new recruits into several different taxonomic groups, and then connect those data to the environmental observations. The goal of all of this is to understand in the long term whether trends in water temperature, acidity, or other parameters are going to change the timing or the success of shellfish reproduction. If waters are warming earlier, we might see the phenology of these species shift. And if conditions become unfavorable, we might see failure to recruit for sensitive animals. This is a photo of some of our great site guardians collecting measurements of eelgrass. And again, we want to connect these biological observations to the chemical observations. So we want to understand whether eelgrass in the different forms that it takes in the Pacific Northwest has different effects on water chemistry. Because for anyone who has experience working with eelgrass, within just the different bays of Washington State, it varies tremendously along a couple different axes. It can range from being almost 2 meters long and very sparsely spread across an environment to being only 10 centimeters long and super dense. And we want to know whether those different types of grass are doing anything different to local conditions. And then finally, these are not ongoing efforts, but we have limited term field experiments that site guardians participate in as they're available. So I'm going to go in detail to the project that these two site guardians are working on later in the presentation. But altogether, the site guardians are helping us collect great data that is being used for advancing the state of the field and for natural resource management, and they're serving as ambassadors to their local communities on issues of acidification and climate change in the marine environment. So just to speak briefly to the scientific output of the network, it's taken a little while to get all of these different parts, the volunteer program, the sensors, the biological observations going. But over the past, it's just been about 12 months, we've had 6 papers published that have been based on anemone. And this is one of the goals of the program that's very important to me, which is to have the network serve as a resource for practitioners, for academic and agency scientists throughout Washington State and beyond, to use for answering targeted questions. Because we're doing the exact same thing across these 10 different sites, it's a really good resource for replicated experiments, because you can pick the sites that are most pertinent to your particular question and then go out, set up the experiment, and know that environmental data are being recorded in the background. So these collaborators who have worked across anemone have asked a bunch of really different questions, from practical efforts to figure out whether putting shell hash into an intertidal environment improves local chemistry, to broad questions about how eelgrass functions across an estuarine gradient in Willapa Bay, to very recent studies that we've just submitted looking at intergenerational effects of ocean acidification stress in calcifying animals, and on curious and potentially important effect that seagrass is having in mediating harmful algal blooms. All right, and just to visualize the amount of data that have been collected through this program, our first sites were established in 2015. Cherry Point was one of those. And here I've just shown within Cherry Point, we've been collecting data inside of eelgrass, represented by the green line, and outside of eelgrass, an unvegetated area represented by the brown line. And each of those black points is a time when we've gone out to swap the sensors. So none of these instruments have any radio telemetry. For us to get the data, we have to actually walk out at low tide, pick up the sensors on that PVC frame, and then bring them back to the lab, download them, recalibrate them, and get fresh ones out. So you can see that at Cherry Point, we've had many different swaps over the past 4 years. There have been a couple times where there are gaps in coverage, and that's because we pulled the sensors out over the winter to avoid storm damage. But across all of our sites, there has been a lot of legwork, and this has been both accomplished by DNR staff and by site guardians conducting swaps across many different sites. And you can also see that there have been different permutations of our sites. We're up to 10, but the Elliott Bay and Squim Bay sites are very recent for us. We established them only at the beginning of this year in partnership with the Port of Seattle at Elliott Bay and in partnership with the Jamestown Squaw Tribe at Squim Bay. So we're excited now to have these new sites where we're looking at an urban environment, very different from anywhere else we're taking measurements, and out on the peninsula in Squim Bay, which is, again, a place where we had no coverage previously. And I hope this just illustrates how we get our data in blocks. Whenever these sensors come back to us, we suddenly have this big multi-month record of observations. And that's the structure of the data as it comes to us. So what do the data look like? I just wanted to briefly show some of the pH acidity data that we've been collecting over the past, this is from the beginning of 2019. And here, there's a couple different things that you can take from this slide. I want to point out that the green box plot is missing for Fidalgo Bay, which is FB, and for Skokomish, which is SK. And that's because the sensors failed at those sites. And this is something that happens with some frequency. And it can be due to a bunch of different causes. So it's another reason that we benefit from having a lot of sites, because occasional sensor failures mean that we're still generally able to answer our big picture questions. And some of those questions are becoming clearer over time, the answers. We can see just from looking at these data collected over the past 8 months or so, that pH does differ between sites. We're looking at places all the way from the outer coast in Willapa Bay to South Puget Sound in Case Inlet, that's on the left there, that's CI, and up into the very northern parts of Washington State at Cherry Point with the CB codes. One thing that we also see that's represented in this figure and in other data that we've collected is that freshwater has really big effects on pH in nearshore environments. This makes sense because the mass of the water over these shallow intertidal areas is not that big. So freshwater, when it's running into them, is going to have more of an effect. But here, if you look at the difference between NRB and the one that is mislabeled right next to that, which should be NRE, the brown and the green plots for NR are very different from each other. And that's because the Nisqually Reach unvegetated spot has substantial freshwater influence that is pushing the pH far down. And then finally, you can see that from the most recent data we've collected, there is not a consistent effect of eelgrass across all the different sites. So when we're just looking at all of the data, the tens of thousands of data points that this figure represents, there is not just a net boost to pH inside of seagrass habitat. But if we look at different timescales and don't just compress all of our data into a single 8-month block, we can come to a finer grained understanding of the influence of seagrass on local chemistry. So here we're looking at the mean dissolved oxygen concentration in one particular place. This is in Case Inlet. And this is looking at over the course of a single month in the summer, what an average day consists of for dissolved oxygen inside and outside of seagrass. So the gray portions of this plot are when the sun is set at nighttime, and the yellow portion in the middle is when the sun is up and there's daylight shining. And I'm just going to show you inside and outside of that daylight block what the course of the day in dissolved oxygen measurements looks like. So in Case Inlet, where there's no eelgrass, you see this characteristic pattern that dissolved oxygen is relatively low during the hours when sunlight is not shining. It goes up as soon as the sun rises and then goes back down in the middle of the day to mid-afternoon. And this is very likely driven by biological activity, by photosynthesis of phytoplankton. And it's this bell-shaped curve that we see over and over again. And in an eelgrass habitat, just 150 meters away from the unvegetated area, we see the same bell-shaped pattern, but the magnitude is quite different. So again, the dissolved oxygen starts to go up in daylight, but it reaches much higher peaks inside of seagrass. And that makes sense because the biomass of primary producers in these seagrass meadows is much higher. And you can see this in the signature of oxygen that's being evolved through photosynthesis. So we see similar patterns to this if you look on just a single day or several day scale for pH, because dissolved oxygen and pH are following the same trajectory. As CO2 is consumed and O2 is produced, the pH is going up and the waters are becoming less acidic and potentially more favorable for shell building animals. So this is just to say that if you look at particular time scales and in particular places, you can see instances where eelgrass activity is driving the local conditions in dissolved oxygen and in pH. Eelgrass has no effect on temperature, which is reassuring because we wouldn't expect it to. And nor does it have any effect on salinity. So this really is an effect of photosynthesis as far as we can tell. And that's probably a combination of both the photosynthesis by the eelgrass itself and by the community of primary producers that adheres to eelgrass. There's a lot of diatoms and epiphytic algae that can grow in seagrass meadows that are probably contributing to net photosynthesis. And when we look at temperature, just because we haven't talked about that phenomena, we can compare different sites in order to understand risks in the future that environmental change might point to. So here in Fidalgo Bay, we're about to look at the average temperature in every month. Each of these pie slices coming out from the center is an average measurement for 1 month. The single blue line there is average temperature in August of 2015. And because we've been taking measurements for several years now, we're starting to get to the point where we can look for trends over time. Nearshore habitats, like all ecosystems, are highly variable, and it's going to take years before we're able to statistically pull apart this variation to the extent that we'll be able to clearly point to the effect of acidification or climate change. But as we move forward, we're getting closer to that point. So here, I'm just visualizing what the 4 to 5 years of measurements that we've taken so far at Fidalgo Bay look like, where the older data is in dark blue and the newer data transitions to green. Overall, there is not a trend over these 4 years in the waters of Fidalgo Bay becoming warmer. But over time, if the water does warm up, we're going to see this continuing pattern of the circle growing outwards, spiraling out to higher temperatures. And if we compare that to another site, this is Willapa Bay out on the Washington coast, we can see that the sites are just different from each other on a baseline. That Willapa is warmer than Fidalgo Bay, and that this probably is going to have biological consequences and present different risks into the future. So what might those risks be? One of them is that on the axis of both temperature and acidity, it's quite possible that as nearshore systems approach critical thresholds that matter to nearshore organisms, that shifts in species composition might follow. And one example of that is the difference in reproductive output by Pacific and Olympia oysters. So Pacific oysters will only move into reproduction and gametogenesis at higher water temperatures. And that's, roughly a threshold of 20 degrees, 19 to 20 degrees Celsius. And as soon as that threshold is hit, they will spawn, and they are more likely to result in natural set in the intertidal environment. So we can see that Fidalgo Bay and Willapa Bay are in different positions relative to that threshold. That Fidalgo Bay still has several degrees of warming before it will hit a point where Pacific oysters are able to consistently reproduce, whereas Willapa Bay is almost already there. And this has been borne out in our shellfish recruitment data that site guardians and DNR staff are collecting, where we see in the late summer, Pacific oysters are recruiting onto those ceramic tiles. So this is one of those places where the biological data that we're collecting through the settlement tiles is connecting to the environmental observations that we're getting through the sensors. And we are ready to see whether in the future, as water warms, there's going to be a shift from potentially Olympia oyster reproduction to more Pacific oyster reproduction. And if we'll see that, it will be in the new recruits on these tiles. All right, so again, going forward and trying to connect the water chemistry that we're measuring through the sensors to the performance of CO2 sensitive animals. Here we have an oyster in the middle of a seagrass meadow, and you can see some bubbles along the seagrass blades, which are a product of photosynthesis. That's oxygen just piling up on those blades and ready to bubble away. I'm gonna summarize some of the research that we've done on the effects of particular habitats on seagrass, or the protected particular habitat on bivalves, calcifying organisms. So All of this work has taken the same general scheme, where we have gotten shellfish that were reared in a hatchery. So they're all closely related to each other and grew up in the same environment. Mostly they were fed as much as they needed to eat. And then we divided those animals and put half of them inside of seagrass stations at our anemone sites and half of them outside, and left them in those conditions for different amounts of time, depending on the species and how fast they grow. But after those exposure periods, we collect the animals and measure a variety of things about them. And this has ranged from the most simple measurements of shell length, where we can see how much they grew over the period of exposure, to detailed studies of their patterns of gene expression across these different habitats, their shell strength, which we've tested through biomechanical methods, and the stable isotope and fatty acid signatures in the tissues of the animals to try and better understand what is different between these different habitats for oysters. As of now, we've done these types of experiments for 4 different species: the native Olympia oyster, the non-native Pacific oyster, manila clams, and geoducks. And even though the particulars of these experiments were all different, I'm going to try and put them into the same categorization to just summarize how they responded to this exposure experiment. So we first conducted work like this in 2016, and then we are again doing work like this summer that is only now taking place. And Olympia oysters, almost every time that we've put them out into this setting, have done better in seagrass than outside of it. So that has generally amounted to a 25 to 35% faster growth rate in shell for the Olympia oysters. In 2018, we found equivocal results, but the other 3 years, we found this enhanced growth inside of seagrass canopy. For Pacific oysters, we conducted pretty broad experiments in 2016 and 17, putting the animals inside and outside of seagrass. And we were surprised at how consistent the benefit was across species, because again, the Pacific oysters grew about 30% faster inside of seagrass versus their siblings outside of seagrass. Manila clams, we've only done 1 experiment with in 2016, and they grew more slowly. They performed worse in seagrass. And geoducks, both times that we have put them out into an experiment like this, have shown no response. They have grown and performed at the exact same rates, regardless of what habitat they're in. So what might divide these 2 groups? For Olympia oysters and Pacific oysters, we've seen better performance inside of seagrass. And for manila clams and geoducks, we've seen either worse performance or no difference. And right here, there is one fundamental difference for anyone who works with shellfish between these 2 groups, and that is that Olympia oysters and Pacific oysters are above ground shellfish, whereas manila clams and geoducks are in faunal and live within the sediment. So they are probably experiencing really different conditions, and the effect that seagrass has may be relevant to one group and not to the other. What do we know about the different conditions that they're experiencing? From our own measurements with the anemone sensors, we can say generally that above ground shellfish are facing pH conditions between 7.7 and 8.1. There are definitely exceptions to this in places where there's really intense freshwater input or particular periods of high photosynthesis where the pH goes above 8.1 in big seagrass meadows. But for manila clams and geoducks, they are not experiencing that water above ground. Their shell and the tissue around the shell is in a very different environment in the pore water underground, where circulation is much slower or absent, and in general, pH is much lower. So there's limited work on the pH environment below ground, but looking back 10 years, there was a fairly good study taking depth profiles of pH, which we can see in the middle here. And what you see is that below the first few centimeters of the sediment surface, pH declines to values that would be extremely low for the water column, down around 7.1 to 7.3. And those are generally anoxic sediments where microbial respiration is consuming all the oxygen, producing a lot of CO2, and producing these conditions that are nearly truly acidic. And manila clams and geoducks, depending on how far they're buried in the substrate, and depending on the particulars of that substrate, are likely running into conditions within this range of 7.1 to 7.7. And they are also clearly able to make it work. They survive in these anoxic sediments under many circumstances and continue to put on shell mass as they grow. So the biological pathways of shell formation between these groups might differ just based on the habitat that they occupy out in the nearshore environment, whether they're above ground or below ground. I did want to mention that one of the studies that's taken place across the anemone network, Courtney Griner's work, also took measurements of pH in pore water and corroborated what the earlier paper found, that the conditions at about the depth where manila clams live are down around 7.1 to 7.4 in pH. So this might explain why the two groups of species here are responding differently to habitat. And then I wanted to dive into, with some specificity, a single one of these studies and talk about how the infrastructure of anemone helps us peel apart the different things that may be influencing oyster performance. So in 2019, we have started a new effort to understand how Olympia oysters respond to seagrass and respond to environmental variation. And the goal of this is to improve restoration outcomes primarily, because right now we don't know enough about what governs the success of restoration actions. And we are hoping to use the natural variation that occurs across the 10 anemone sites to test whether Olympia oysters perform better, survive better in warmer water, colder water, more acidic, less acidic water. And we'll know what those conditions are because of the sensor observations. So this was again accomplished through taking hatchery animals, in this case from the Puget Sound Restoration Fund, Manchester Hatchery, and sending them out to all 10 of our anemone sites. And at every one of these sites, we put them inside and outside of seagrass. So this is our most replicated effort yet. It includes the two new sites at Elliott Bay and Squim Bay. And at about monthly frequencies, DNR staff and site guardians have been measuring the growth of these animals so that we can generate growth curves and understand whether particular stressful periods are producing mortality events. Rather than just taking beginning and end measurements, we decided that it was important to get as much data as we could in the middle. So what are we finding here in broad terms? This is still underway, and in about a month, the experiment will come to an end. We're going to collect the animals from all of the 10 sites, bring them into the lab, take final measurements of their shells to see how much they've grown and how well they've survived. And then we're also going to dissect them and look at patterns of gene expression and at their microbiome, at what they're ingesting, and we're also hoping to look at whether they're incorporating potentially harmful algae into their diet. So this is what it looks like in the field. This is an important caveat for all of the transplant work that we've done with shellfish. You can see here in the middle of the bottom of this photo, there's a PVC structure with these two mesh cages. And for every one of these experiments, we've put the shellfish out inside of predator exclosures. So we really are just looking at the influence of water conditions, which include food availability on shellfish, and we're preventing predators from having any access to the animals. So I just want to say that it's possible that predator dynamics or interspecies interactions may dominate the signals that we're seeing in the real world, because it's totally possible that Pacific and Olympia oysters do better in seagrass because of some chemical phenomenon, but it could also be possible that their predators are more prevalent in seagrass, and in the real world, they just get eaten and don't get a chance to put on more shell mass. So this summer, across the 10 sites where we put Olympia oysters, I'm going to show you how their shells have changed in length, how much they've grown as a percentage of their initial length. So in unvegetated areas, we've seen over a pretty long period, it's been about 200 days since we put them out in January, about a 4% increase in shell length over that time. So we knew from some previous work that Olympia oysters are very slow growing, so we had to plan for a long exposure period. We can pull off these experiments much more quickly with Pacific oysters just because they respond so fast to the environment. And in 2019, we're seeing something similar to what we found in the past, that there is just a boost in growth inside of eelgrass. So here are the measurements as of 2 weeks ago, I believe. And we're finding that Olympia oysters are growing faster in eelgrass yet again. So this helps us add to the body of evidence that habitat matters for these CO2 sensitive animals. But I wanted to delve a little bit into the sensor measurements to show how we are able to pull apart what the mechanism might be behind this growth difference. So inside of seagrass, you would expect there to be some differences in environmental conditions. Like we've already seen, there can be transient differences in dissolved oxygen when the grass is photosynthesizing in the middle of the day. Those differences also are a mirror of pH conditions, whereas temperature and salinity are not likely to be different. And because we have these two habitats at many different sites, we're also able to take advantage of the natural variation in environmental conditions across the sites to look at, besides habitat, what drives oyster performance. So here, I'm going to show you how a couple different environmental predictors connect with oyster performance or don't. So for the 3 months after transplanting the Olympia oysters into the environment, I'm showing the average temperature on the x-axis here and the percent change in shell length on the y-axis to see whether the average temperature controls growth. So here are the outcomes for oysters across many of the different sites. And you can see just with these points that overall the animals in eelgrass were growing faster. They're higher up on the y-axis, but there does not appear to be any connection, and there's no correlation between temperature, average temperature, and shell growth. So that does not appear to be one of the mechanisms that's driving growth patterns. So I'll exit out. What about extreme temperatures? There's a lot of different ways to look at this, from the variation that's experienced by the animals to the incidence of harsh episodes. And here I'm taking the latter approach. So on the x-axis, we have the number of temperature observations above 15 degrees Celsius. And that's really just an arbitrary threshold. I don't know whether it's particularly stressful for these animals. But here again, we see that there's not a strong connection between the number of times the animal experienced conditions above 15 degrees and the amount of growth that they achieved. So temperature does not appear to be connected to this growth pattern. For average dissolved oxygen, we again just see a mishmash of points that doesn't help us explain the change in shell length. And when we look at extreme dissolved oxygen events, here I've just taken the threshold of hypoxia, so the number of observations below 2 milligrams per liter. And we do see something kind of interesting, which is that across our many sites, there were 2 sites that experienced some amount of hypoxia for a while. So there were about between 500 and 600 observations that were below this threshold. And those also happen to be some of the lower performing oysters, the lower growing ones. So here, the evidence is limited, but there might be some connection between extreme dissolved oxygen conditions and shell growth in Olympia oysters. All right, so what about pH and acidification risk to these animals? The average pH conditions across all the sites are here on the x-axis, and again, the change in shell length on the y. And here, this actually does appear to explain some amount of the variation in shell growth, where we see that the sites that have on average higher pH have a bit higher growth. So this is one point in favor of the hypothesis that eelgrass produces higher pH conditions, which then translates to better performance for the oysters. And what about extreme pH? Here again is just an arbitrary threshold, the number of observations below a pH of 7.6. And again, it's hard to make very much of this pattern because we have one site that was a big outlier where there was a period of low pH. And there does not appear to be a connection from the data set that we have so far. So looking at these 6 different causes, this is one example of how we can connect these community science efforts. The oysters are being measured by site guardians across the network with the sensor observations to figure out what is it that is driving differences in growth for shell-building animals and how that relates to eelgrass versus no eelgrass habitat. So there's been a lot of work that's occurred at Anemone on different topics across the 4 years of its existence. Some of them I've already mentioned about whether shell hash is a practical management option to ameliorate chemistry in nearshore environments, to other studies of gene expression and stress signatures in shellfish. But in this talk, I just wanted to focus on the efforts that we're doing year over year to understand shellfish and particularly commercial shellfish performance inside and outside of seagrass. And we're still trying to understand this benefit that Pacific and Olympia oysters appear to get from association with seagrass. We've had mixed evidence over the years. This latest experiment, as we just went over, appears to offer some support for the idea that it's pH conditions and acidification environment that matters for these animals because the faster growing ones are connected with a higher average pH. In other cases, we haven't seen that connection. And I think the growth benefit probably has more to do with the food availability and the food environment inside of seagrass. So with that, that is a summary of the work that we're doing in Anemone, the Acidification Nearshore Monitoring Network. And if anyone in the webinar is interested in engaging with the program and being a part of these ongoing chemical and biological measurements and participating in field experiments, you should contact Erica Blakey, who I think is on the webinar, who's our volunteer coordinator for the program. And next year, we're looking forward to having our biggest cohort of site guardians and picking their brains for ideas on experiments to execute across Anemone to make sure that the work that we're doing is relevant to growers and just interested community members across Washington State. And I'll close with acknowledging the many, many partners that have helped out with this work over the years. Okay, I think there's time for questions. And if Terry King can come back on and help out with that, we'll see.
I am back. I'm sorry I couldn't find myself in the extended list of all the participants, so thank you for bearing with me. Micah, thank you for your presentation, and we do have time for questions. So there's a Zoom group chat box that you should see on your webinar, and if you want to send a note privately, that would be great. We do have one question, and that is, it's important to know which sensors actually work in the field. Would you be willing to talk about the sensors that you used? And before you answer that question, I want to remind folks that we do have on the California Current Acidification Network, and that's c-can.info, we do have some guidelines and some best practices that were developed by the California Current Acidification Network. And so you might also find those helpful as you work to select your gear. So Micah.
Sure, I'm happy to talk about sensors. For dissolved oxygen measurements, we use a combination of onset HOBO sensors and PME mini dot sensors. For chlorophyll, we also use PME Cyclops 7 sensors. For salinity, and we use conductivity and temperature loggers from Odyssey, a New Zealand company. And for pH, we build our own sensors and they are effectively a prototype version of the commercially available Satlantic CFAT sensor.
That's great. So we have another question coming in from Megan Hines, and it is for the 2019 Olympia Oyster Project. Can you remind me when the oysters were outplanted and how long you measured growth for? And then she's also wondering how different the data might look after the summer when the growth rates are at their highest.
Yeah, that's a great question. We put out the oysters at the same time across all the 10 sites, and it was in the middle of January 2019. So they've been out there not quite 7 months, but getting there. And the whole outplant period is going to be only another month from now. We're going to collect them at the end of August, beginning of September. And we have definitely seen differences in growth over the seasons that they've been exposed in the intertidal environment. That at the beginning, when we were collecting these monthly interim growth measurements, there was really no change, probably between January and May. And then from May until now is when pretty much all of the growth has happened in the warmer waters and higher productivity that they're experiencing now.
Great. Anyone else have a question? Oh, from Caitlin Magel, I hope I got that right. Have you noticed seasonal differences in the effect of eelgrass on water chemistry?
Yeah, that is a great question. The figure that I showed with sort of an average day in seagrass for dissolved oxygen, where we saw the bell-shaped curve outside of grass and then the much higher bell-shaped curve in grass, that took place right in the height of summer, I think in early July in Case Inlet. And if you look at if you tried to generate that exact same set of data in December, it would look very different. So we do see that in the winter, there is a negligible signature of photosynthesis from seagrass. In general, the plant has a lot of biological timing to its lifestyle. In the winter, it shrinks in size, it becomes much less productive, and in some cases almost stops photosynthesizing altogether. And then in the spring and summer, it gets much, much bigger. It starts photosynthesizing more rapidly, and we can see that difference in the water chemistry.
Great. So questions are rolling in. Celeste is interested to find out, how do you ensure the safety of your sensors when deployed in the field?
Yeah, we just cross our fingers. We have them secured to augers, screw anchors that are in the substrate. So that has so far been successful in keeping them safe from waves and any stray debris. We've been very lucky in not having any or very minimal human interference. Nobody has just come across one of our sensors and taken it away. That did happen once, but the person just gave it back to us. They picked it up because they thought it was debris. So we don't have any measures in place really besides labeling all the sensors with my cell phone number. So that was on one of the early slides, and anybody who comes across them, I get calls, especially during the summer, from beachcombers who come across the instruments and just call and say, hey, what is this PVC package with all these pipe bomb looking things attached to it?
Great. So Lilia is wondering if there are any results or observations from the chlorophyll monitoring at Cherry Point.
Oh, off the top of my head, I don't know about that, actually. We are collecting the measurements there, but yeah, I'm sorry that I don't have a memory of how Cherry Point compares to other places.
Okay, great. And then Erica just let us know that another question is about, relative to Cherry Point, is how is the barnacle incrustation affecting Olympia oyster growth? And has it been more apparent at the bare sites across Anemone?
Yeah, this is fairly anecdotal. We're relying on our site guardians when they go out to take field notes and let us know what's happening when we can't be there. But we have seen a pattern this year, which has seemed to be the same in the past few years, that barnacle recruitment is much greater outside of seagrass than it is inside. So that has implications for the reliability of sensor measurements, because it means that it's more likely for biofouling interference to happen outside of seagrass. So in the past, before we had our great cohort of site guardians, we would often go and pick up instruments after a long deployment and find out that barnacles had started to grow directly on the sensing element of the pH sensor, the dissolved oxygen sensor. So we were measuring conditions inside of the barnacle shell. And now that we have site guardians, that has become much less common. It's easy to catch barnacles early in their life and just wipe them off with a cloth. But it seems to be necessary more frequently outside of seagrass that the barnacles are glomming onto our sensors there more often.
Mm-hmm. Great. That sounds wonderful. So another question from Lilia is, how do you determine the number of Olympia oysters to put in each satchel? And do you perform statistical power calculations ahead of time to determine that?
We decided this year, all of our choices this year were really governed by past work that we've done with Olympia oysters. So I already spoke about how we knew that we had to have a much longer exposure period for these animals because of how slowly they grow. We also had data from past years about their mortality rates. So we tried to pick a number where, based on our past observations, there would still be a majority of the animals living in every cage at the end of the summer. So that was just looking back at past life and death measurements that we took of the animals. I did do power analysis on the whole experiment, and that mostly was taking into account the number of sites that we were using for the effort, because we are not counting the individual oysters inside of those cages as independent replicates. All of those data that I showed you about the average change in shell length were sort of agglomerated. That's the average, the change in the average shell length within a cage. Because all the animals are living in the same cage close to each other, experiencing the same conditions, we're not treating them separately.
Great. And one last question from Diane is, do you study invasive seagrass beds?
We do not. So far, all of our stations, the eelgrass stations across the network, are in the native grass zoster marina. Maybe I should have mentioned that. And at the tidal depth that we work at, from -1 to -3, that's the predominant grass that you'll find in Washington State. If we wanted to study zoster japonica, the non-native grass, we would have to establish stations further up the beach and probably look to some different places, because not all of our 10 sites have zoster japonica present. As far as I know, there hasn't been work done on the effects of zoster japonica on water chemistry in the field. I would expect the effects would be very similar to zoster marina, because the per area productivity for both species is very close to one another. So the amount of CO2 that's being soaked up and the effect on pH, I think, is likely to be similar.
Great, thank you. Well, I think that's the end of our questions, and I just want to let everyone know, oh, wait, one more question I will do. Lori says, thank you for the nice talk. You mentioned that temperature may play a role in recruitment success of oysters and could affect oyster variety composition as water warms. Have you looked at other organisms that might be competitors to the oysters?
No, we haven't. We are looking at barnacles. Whenever we pull our recruitment tiles out of the water, we're doing counts of how many and how frequently you see barnacles. But if there are other target organisms that we should be looking for that are sensitive to particular temperature thresholds, I would love to know more about them. We are really eager to add more to our existing biological observation protocols.
Great, wonderful. So that leads to, if anybody's interested in reaching out to Micah directly, I've put his email address into the chat box, so we've got that. A video recording of this session will be shortly available on the California Current Acidification website. You can also join the CCAN listserv on the website, where you can also find out about CCAN activities and future webinars. You should also check out the Ocean Acidification Information Exchange and join the CCAN section. Thank you for attending the CCAN Ocean Acidification Roundtable today. We welcome any feedback, further suggestions for topics in this webinar series, and you can submit them in, your information or your suggestions in writing by emailing us at the address on the page, which is Diane. We're really pleased to announce our next discussion will be on Wednesday, September 18th at 1 o'clock. And this is our normal schedule for the acidification networks. We generally do it on the third Wednesday. And it is by Dave Hutchins from the University of Southern California, Los Angeles. And the title of his talk is How Ocean Acidification Works Hand in Hand with Warming and Other Global Change Stressors to Promote Toxic Pseudonychia Harmful Algal Blooms Along the West Coast. So I want to thank you, Micah, for your presentation today and sharing your expertise and perspectives. And I really want to thank all of those site guardians that I know are on this call today and everyone else involved with Anemone and everyone that's interested in learning more for joining us and sharing this great knowledge. So thank you very much, and I'll end the session now.